Hi guys, this is another video which is the second video where it's quite planned and not impromptu. And today we are going to discuss about influenza disease and which is of a respiratory tract disease and which is caused by influenza virus. Here some few important details are going to be discussed. Amongst those are morphology of the influenza virus, classification and antigenic subtypes of influenza virus, antigenic variations of virus in which we have antigenic drift and antigenic shift, mechanism and significance of antigenic shift, also with pathogenesis of influenza virus, and if time allows we'll also dip into the lab diagnosis of influenza virus. So now let's first begin with the influenza virus. So this is the pictorial representation of influenza virus, and you can see this is the virus which causes an influenza disease, and they belong to the member of family known as Orthomyxidae family, and they are the reasons of quite of morbidity and mortality. Influenza virus consists of 4 genera. Their classification will be discussed later, but first let's see about the morphology of influenza virus. So the influenza virus is grossly 80 to 120 nanometer in size, and it has a helical symmetry because we say that it has a helical nucleocapsid. This entire blue matrix protein or uska inside ka part is called nucleocapsid. This is helical nucleocapsid which is surrounded by an envelope. Let's see it in more manual phase. So discussing about influenza virus. Influenza virus, as discussed, it is 80 to 120 nanometer in size and it shows a spherical symmetry. It contains, so first let us draw the virus. So it contains of an envelope, so that's the envelope of the virus. So I'm drawing a lipid envelope, and inside a lipid envelope there is a nucleocapsid. So first let's discuss about a nucleocapsid. Nucleocapsid contains a basic thing which is called matrix protein. So these are the matrix proteins of the nucleocapsid. So inside the matrix protein we'll find multiple segment of negative sense single-stranded RNA. So multiple negative sense single-stranded RNA ka multiple segments. So these are the multiple segment of negative sense single-stranded RNA. Now each segment codes for viral proteins. So what are viral proteins we have? Basically we have 8 structural proteins and we have 2 non-structural proteins. Okay, so we will see what is basic site of replication. Site of replication for all other viruses is in the cytoplasm, but here the site of replication is in the nucleus. So we have 4 genera of influenza virus. We have genera A, B, C and D, and amongst this genera A and B consist of in total 8 viral protein. C and D consist of in total 7 viral protein. So which is the missing viral protein? Okay, it will be understood later on. So viral proteins, in which we have 8 structural proteins as discussed earlier, in which we have 8 structural proteins. Those 8 structural proteins mean first is the PB1, PB2 and PA protein. So these 3 are the polymerase protein which are responsible for RNA transcription and RNA replication. Both of the process takes place in the nucleus. So these polymerase proteins, so they come under the class of polymerase proteins which are responsible for RNA transcription and RNA replication. Next set of proteins is nucleoprotein known as NP. As the name suggests, nucleoprotein, they will be forming they are the capsid proteins, and those capsid protein will be forming the nucleocapsid. So yes, these are the main, which shows the helical symmetry. So yes, NP protein, so in the nucleocapsid protein. So entire nucleocapsid form करने वाला capsid protein. So we discuss up till now 4. Rest 4 proteins में we have M1 and M2 proteins. Now understand this M1 and M2 protein. These proteins are also called as the matrix protein because they code or the matrix virus. So matrix protein, they will be lying underline the envelope. So this is the envelope also known as the lipid envelope, and it is lined by matrix protein M1 and M2. So matrix protein M1 lies what beneath. So it is this is known as a matrix protein which forms the outer layer of the nucleocapsid, which is lined by M1. And next is M2, and this M2 is responsible for forming ion channels in the envelope, which helps in transport of molecules. So such ion channels are formed like this. I'll show. Yes. So these ion channels form which helps in transport of molecules. So for the transport of molecules, M2 is there, and this matrix protein is M1. So now by now you have got to know about 6 proteins. Now the next proteins are known as non-structural proteins, which we have NS1 and NS2. I know 2 more proteins of structure but are remaining, but first let us discuss about the non-structural protein, in which we have NS1 and NS2. Why they are called as non-structural? Because their action is functional action, therefore they are called as a non-structural proteins. What is the functional action in which we have two? We have heme agglutinin and we have neuraminidase. So heme agglutinin and neuraminidase. Now understand this heme agglutinin protein, what it will do. It is basically a triangular shaped peplomer, and it will bind to mucin and sialic acid residues on the respiratory epithelial cells, and it will cause the facilitation of the entry of virus. So this is a triangular peplomer. So it's like this, I'll show. So this is triangular peplomer known as a hemagglutinin. Hemagglutinin triangular peplomer. It will attach to mucin or sialic acid receptors on respiratory epithelial cells, and this will cause entry of virus. So basically, yes. Now next is the mushroom shaped cells, which are known as a neuraminidase. This is sialidase enzyme. Once it binds to sialic acid receptor, this acts as a sialidase. As the name suggests, dase, it will break down the sialin, and it will cause the facilitate the viral entry entirely from the infected cell surfaces during budding process by preventing self-aggregation of virions to host cells. Means what it does, that neuraminidase will facilitate virus particle from infected cell surfaces during the budding process by preventing the self-aggregation of virion to host cell. Now understand, this is the respiratory epithelial cell. Now this respiratory epithelial cell में the virus has entered by the action of heme agglutinin. Now these are the virion particles. Now this virion particle will undergo replication by the process of budding. Now this respiratory epithelial cell, after entry of the virion particles, is now called as an infected cell. So infected cell surface में they will undergo budding. Now this budding can also result in self-aggregation of the virion particle. So in order to avoid this self-aggregation of virion particle, the sialidase acting enzyme known as neuraminidase protein is being produced, and thus it passes through the mucin layer or sialic acid layer in the respiratory tract. It causes and thus reach the target epithelial cell. So there are two important action of neuraminidase. So now this 2 is done, NS1 is done, NS2 is done, non-structural protein, and this is known as envelope protein. Now let's come to NS1 and NS2, which we discussed earlier. These were about HA and neuraminidase. Now we will go for the NS1. NS1 stands for basically antagonist of interferon and inhibition of pre-RNA splicing. Interferon is there, which causes RNA. Interferon causes RNA splicing, also known as a pre-mRNA splicing. So ये process रुकने के लिए NS1 is responsible, and NS2 is responsible for export of molecule across the nucleus, as this what known as Na. It forms a channel for M2 M2, which forms the channel in the envelope for transport of molecule. This NS2 is there, which causes the transport of molecule, and N1 stops the interferon, thus prevents the mRNA splicing. Again, important thing, neuraminidase, two important action of neuraminidase, that is silidase action. I told you, once it enters a respiratory cell, it becomes the infected cell, undergo budding as a form of replication. It may cause the aggregation of the virion particle. So to avoid the aggregation, self aggregation in host cell, it facilitates a release from the infected cell surface, and thus it can infect more number of cells. And also it helps to pass the mucin. Mucin is a protective layer in the respiratory tract. So neuraminidase, it will just break down the neuramin bonds in mucin, and it will just help to pass to the respiratory tract layer. So yes, that was about the morphology of influenza virus. You can have the look, same as structure. Now let's come to a new topic, which is antigenic subtype and classification. In this topic, subtype means, as we discussed, it had 4 genera A, B, C and D. Now let us understand the subtypes, क्योंकि genre के भी subtypes होते हैं. So subtypes में we have HA and NA antigens. Based on these two antigens, we have discussed the subtypes, HA antigen and NA antigen. So influenza A has flu A, will write, has 18 H subtypes, 11 N subtypes. That is, it has 18 antigenic variants and 11 N antigenic variants. And also most of the subtypes infect animals and bird, but undergo genetic changes and infect human. And these are the subtypes: influenza virus, HN, we write H and N, so which caused epidemic, H11 N11 something. So what does it indicate? It indicate इतने 11 सबटाइप में से ये वाला सबटाइप वेरिएंट और 18 वेरिएंट में से ए वेरिएंट. So that antigenic variance is there, so vast. So 18 H and 11 N subtypes, ठीक है, and these are responsible for basically major epidemics and major pandemics. For example, 6 HA and 2 NA subtypes have been recovered from human, and rest subtypes have been recovered from animals. Next is influenza B. Now this influenza B are not classified into subtype but have diverged into lineages. Currently circulating influenza type B virus belong to either Yamagata or Victoria lineage. So we have two lineage. We have Yamagata lineage and we have Victoria lineage. So these two lineages are common. Next is influenza C virus is detected less frequently and usually caused mild infections, does not present public health importance. And influenza D virus, it causes primarily affect cattle and not pathogenic to humans. So both of them safe. B is very much dangerous, and A is dangerous. 18 as subtypes and 11 and subtype based on HA and NA antigens. So the standard nomenclature of system for influenza virus, ठीक है, human strain influenza A, Hong Kong 03 1968, non-human strain swine. Okay, so these are the standard nomenclature. Now you can see the standard nomenclature over here. Influenza A, Hong Kong 03 1968, swine Lova 1930. Next is antigenic variation. So our next topic is going to be antigenic variation. So antigenic variation is basically a very unique property. So this is a very small topic, antigenic variation. Antigenic variation which is heavily dependent on HA and NA papilloma strains. इसमें कोई mutation होता है और there may be a certain genetic reassortment because of which there is a seen in antigenic variation. First we will discuss about antigenic drift, which is the type 1 antigenic variation. Antigenic So coming on antigenic drift is basically a minor change, also known as point mutation. It occurs either in the peplomer HA or peplomer NA, which result in the alteration of amino acid sequence at the antigenic site of HA or NA. Now understand mutation causes the amino acid sequence change at the antigenic site of HA or NA, such that the virus can partially escape recognition. Antigenic sites at its epitope are the sites of viral recognition for the host cell immune system. और अगर उसी साइट पे there's an amino acid sequence change which results in a point mutation, as a result there's an antigenic alteration, then it becomes really difficult for the immune system to recognize the antigen of the basically virus, and the new variant must be sustained toward more mutation to become an epidemiological significant. Seen in both influenza virus type A and B, results in outbreaks of minor periodic epidemics, and antigenic drift occurs more frequently. It is every 2 to 3 years, but that is not that significant. More significant is antigenic shift. Antigenic shift, in which we have few characteristics of antigenic shift. Antigenic shift मध्ये first we have is that it is major, it is drastic, it is discontinuous. As the previous one occurred like every 2 to 3 years, it is not like that. Discontinuous, and it is basically variation in the sequence of viral surface protein, that is HA or NA. Sequence of virus protein, that is also known as a result of genetic reassortment. What are the characteristics of the genetic reassortment? The new antigenic variant strain is not at all similar to its predecessor strain, previous strains, and it is totally new. It is unrelated antigenetically to the predecessor strain, and infected the same host cell result. What happens in this process is there are two or more influenza virus which infect the same host cells, because of which there is a change in the sequence of viral proteins. As a result, the change is major, drastic, or discontinuous. First, what happened? Two or more viruses, okay, infect the same host cell, because of which there is a change in the sequence of viral protein. This change in sequence of viral protein is what? It is major, drastic, and discontinuous, which will lead to genetic reassortment, and totally unrelated antigenicity will be acquired, and this unrelated antigenicity will lead to a new strain which is completely different from the predecessor strain, and the antibody developed against the previous strain become ineffective. So previous strain के against जो antibodies थे, all of them turn ineffective to them. So this is the mechanism of antigenic shift. It occurs, and talking about the significance, it occurs only in influenza A as compared to antigenic drift, which occurred in both influenza A as well as B. It is pandemic, and it is a major epidemic. Example is H1N1 pandemic 2009. But here in antigenic drift, it is like a major outbreaks and minor epidemic wala part, and it occurs more frequently. It is 2 to 3 years, but this occurs like less frequently, 10 to 20 years frequency. So that is the antigenic variation. Now our next part is pathogenesis, which is actually the last part of this video, guys, because lab diagnosis unfortunately couldn't be covered. Then pathogenesis में first thing is the pathogenesis में what we seen कि how it causes is transmission. Transmission is through the respiratory droplets, or by sneezing, or by coughing. So those people with 1 meter distance, or contact with surfaces of fomite of with respiratory droplets, and then touching nose or eyes. These are the things. Next is target cell entry. Viral HA attached to specific sialic acid receptor on the respiratory mucosa that lead to virus entry. Next, multiply locally. Virus replicate in the infected cell, and infectious daughter virion spread to adjacent respiratory cell over several hours by the action of HA and NA. Then this is target cell entry and multiplication. Then talking about multiplications, spread. Very early virus also spread to lower respiratory tract and spills over bloodstream to involve extrapulmonary site. And next is local damage. Influenza virus infection causes cellular destruction and disformation of superficial mucosa of the respiratory tract. may dispose to or may predispose to secondary bacterial infection. So yes, this is a thing. Transmission, target cell entry, multiplication, spread, and then local damage. Local damage by cellular destruction and local damage by desquamation of superficial mucosa, which lead to secondary bacterial infections. Okay, that was about the pathogenesis. So we discussed basically the virus introduction, morphology, then we discussed about the antigenic subtypes and nomenclature, then we went to antigenic variation, and then pathogenesis of the influenza virus. So basically this was a basic summarized form of influenza virus. So yes guys, I hope you enjoyed the video and you enjoyed learning multiple aspects, though few in number, of influenza virus. These were the really crucial ones and really very much important ones of influenza virus. Yes, and influenza very common viral infection, and उसके बारे में थोड़ा बहुत तो you should know. And yes, this is a good thing that we are ending this video on a nicely explained array. This is more experienced, by the way. Yes, and I hope it helps a lot. It helps a lot in understanding virology, medicine, also pharmacology में when you introduction of drugs, pharmacy, normal microbiology, biology students. For all of these this is a sustained approach and very much simplified manner, very much minimal and very much understandable things have been kept in this video. Again, this is another sincere effort by me, and also it did in turn impromptu only, though it wasn't before. Okay, thanks for watching, and I hope you encourage more to upload more such videos and getting more derived up by the beautiful subject. Yes, thank you.